experiments will complete, and 17th you will be uploading your PDF, single PDF of all the 4 experiments in the Google Classroom, and I have prepared like X1, X2, X3 batch separately. Rooms have been created. Okay, so separately, like where the assignment part will be given, that time you can assignment experiment number 1 to 4 I'll be mentioning, so you can then upload there. Okay, so we come to like before we go to this experiment, can you tell me what we had discussed earlier in the practicals? Yes, anyone?
We have discussed the dissolved oxygen experiment.
Yeah. The first experiment was determination of alkalinity. Second was hardness. Third was dissolved oxygen. Okay. Now the fourth experiment is you can write the aim determination of free chlorine present in the given water sample. And this is also an iodometric method. Yes.
Can we write this now?
Yeah, you can write or like that book PDF, practical book PDF, is it not containing that part which I gave you long back? Chlorine. I gave you practical part, right? Hardness experiment number 2. Okay. Before that, I think I gave you certain. Yeah, here is the book. Wait a minute. Okay, let us see this book PDF. I'll give you PDF of this also. Okay. Okay. So, let me explain the thing. What all things you require is burette with clamp and stand, pipette, conical flask, beaker, etc. Standard N by 40 potassium dichromate solution, potassium iodide solution, uh, sodium bicarbonate is required, then N by 40 sodium thiosulfate solution is made, and a starch solution as an indicator is required. The theory says that free chlorine is never present in raw water or natural water. So chlorine, which is widely used for the disinfection purpose, for making the water potable or drinking quality, or making it capable for drinking. The municipal water supplies treat it to remove bacteria, fungus, and other pathogenic microorganisms, and for deodorization also. So since it is a powerful.
Ma'am, your voice is lagging.
Yeah, I have muted the thing. See, like I was busy sometimes, so like I had muted the this particular meeting. Now, can you hear me?
Yes, ma'am.
Yes, madam.
Yes, ma'am.
Okay. So you have like you want to determine free chlorine. Now free chlorine as such will not be there in any raw or natural water. Now chlorine is added for disinfection purpose so that it is made potable. Potable means it's like biological flora like bacteria, fungus that should be killed, and the water is made potable or of drinking quality. So it is also at the same time it is deodorized also. So odor, if it is there because of the decaying matter and all, that is lost. So since chlorine is a powerful oxidizing agent and is cheaply available, so chlorination is done with the help of bleaching powder or chlorine gas or chlorine dissolved in water in the form of concentrated solution or with chloramines. The sterilizing action of chlorine is supposed to be due to the reaction with water producing hypochlorous acid and nascent oxygen, both of which have powerful germicidal properties. Now, what is saying? This is the bleaching powder. Now, why we are writing with bleaching powder? Because bleaching powder is the cheapest chlorinating agent, and it is seen that when you add bleaching powder you can add specific quantity, like you can weigh it and add so that you can decide upon the dose also. And when it goes into water it forms calcium hydroxide and chlorine gas. Now this chlorine further gives formation of hypochlorous acid HOCl now and HCl. This hypochlorous acid itself is a bactericide. It attacks the cell wall of unicellular or the living organisms. And if the cell wall is ruptured, then eventually the cell or the organelle, the microscopic organism will die. Apart from this, the hypochlorous acid further it gives formation of HCl and nascent oxygen is released. So this nascent oxygen anyhow will oxidize the biological material, and the oxidation product may be CO2 or whatever it is. You will find that this oxidation which happens due to this nascent oxygen will kill the germs. Okay. So however, the excess of free chlorine. Now, what is happening? The chlorine is added. Now, you can add bleaching powder. You may add directly chlorine gas, but adding chlorine gas is expensive because getting chlorine gas is expensive. So that makes the sterilization process expensive. Whereas bleaching powder, yeah, it is like cheaper than your chlorine gas. Another chlorinating agent is chloramine, but in chloramine you don't get formation of chlorine. Okay, so this is how chlorination is performed, or when chlorination is performed, chlorine is incorporated into your, or chlorine is coming into your water. Now, what we are determining is free chlorine. Chlorine, it is not Cl minus. Okay, keep in mind this is Cl2 gas which will be in dissolved condition in water. Okay. So, however, the excess of free chlorine in drinking water is undesirable. So, from where you are getting free chlorine? From the sterilizing agent, that is bleaching powder or chlorine gas. So, this chlorine in drinking water is undesirable. and as it is not only unpleasant for drinking but is also injurious for human metabolism. So, hence, the amount of free chlorine in municipal water is estimated prior to the domestic supply so as to make necessary adjustment in the dose rate accordingly. Like what should be the dose? If excess of chlorine is there, then early morning, if you collect the water supplied to the locality by the municipality will be treating it and sterilizing it. So lot of chlorine smell will be there in the to the water. So that should not be there, or it should be like to certain extent only. So the principle involved in
Ma'am,
determination, yes,
how it is injurious for metabolism? It's chlorine in water.
chlorine in water, like you see, when you consume chlorine, it is or water containing large amount of chlorine. First of all, the effect is like your buccal cavity, the mucous membrane of your buccal cavity, that is attacked. So there it is consumed. Then water containing chlorine, if it is going into your metabolism, then you can expect something like this taking place there also when it goes into the metabolism. So this may be like acting upon the your what all enzymatic action is there. Like you drink water and it goes into the stomach. Now there some mechanism by which it is acting upon the enzymes and all may be there, and that is how it interferes in the metabolism. Now, very exact part I don't know, but two things are important. Firstly, when you drink water which is rich in free chlorine, it attacks the mucous membrane of the buccal cavity. So here only like your mouth and this whole thing you will feel after drinking water. Sometimes you will keep feeling dryness in your mouth. Many a times you must have observed when you like drink some these like carbonated drinks like your Coca-Cola and other things like you must have observed that when you drink such carbonated water or these things, your mouth finds like some dryness. So that is because of the mucous membrane of the buccal cavity that is affected because of either chlorine or even the carbonated water and so on. So similarly, when it goes into the stomach and like there it is reacting with certain enzymes, that is why it is said that it is injurious for human metabolism. Okay. So if you know these two things that it interferes in the human like metabolism, that is sufficient. ओके तो एग्जैक्ट एक्शन वो एटीपी को डिस्ट्रक्ट करता है या एनएडीपी को करता है दैट दैट आई हैव नॉट सीन और इफ यू गेट द थिंग यू लेट मी नो ओके
Okay, ma'am.
it's a clear or not okay
It
so
is, ma'am.
exactly it interferes in the metabolism and the important part is we know that if this part is there it is definitely going to form something like this. Now it depends like what is your substrate inside your stomach or intestine where it is moving, right? So how it goes and binds to which particular site, अब कौन से इसके site को जाके bind करता है, that you can Google. Okay, it is good that you have that curiosity to know how it is interfering in the metabolism. Now the best way is morning में पानी collect करके रखो, collect it in a earthen pot. 2-3 times the earthen pot is opened. So the number of times you open the pot, the drinking water pot, the chlorine tends to escape out. तो शाम तक आपको क्लोरीन का जो कंटेंट है वो स्मेल it reduces, right? So means it is not very stable and does not remain for a long time in water. Like if you pour water from a higher level to lower level, or the water is trickling down, it is seen that the chlorine content will be reduced at the bottom if you pour the water from the top. Okay, so that is how it moves out also easily. But then, yeah, it should not be there. That is there. Hence, so where principle. Okay, so what is the principle involved in determination of this free chlorine? The principle involved is this free chlorine, estimation of free chlorine in water is that when a measured quantity of water is treated with excess potassium iodide, okay, so you measure some quantity of water. Of course, it will be the municipality treated water. If you directly take some well water or like from some pond or river might be containing, but these pond and rainwater directly, you will not get this part. So municipality treated water will be containing chlorine. So this water sample is taken, to this potassium iodide solution is added. So when you add potassium iodide solution, without adding like this Ki contains like when the Ki solution is prepared, 1 ml of concentrated H2SO4 is added to it initially here itself. So here you don't have to separately add any dilute H2SO4. What all H2SO4 is present in this Ki, that it's like there is no need of H2SO4. So this chlorine will be oxidizing the iodide to iodine, and it will liberate equivalent amount of iodine as much of chlorine is present, forming KCl. So chlorine oxidizes iodide to iodine. The quantity of iodine formed is equivalent to the chlorine, free chlorine present in water sample. Now, once iodine is released, now you will see that actually when you perform the experiment, you have to take double the quantity of water sample, because here chlorine is less. So because chlorine is less, we ask students to take double the quantity so that you can get. Now here you don't get very dark brown color because of iodine, because the chlorine is less, so less of iodine is released. So the solution color is yellow only. Okay, so this iodine imparts yellow color to the solution, and this is then first titrated with the sodium thiosulfate solution till you get a pale colored solution, that is the content of iodine is reduced, and you get formation of sodium tetrathionate and twice NaI, and iodine is still there, uh, which imparts a pale yellow color to it. So to this solution, which has now like you have titrated with hypo solution and made it pale yellow, to that pale yellow solution you will add starch indicator. Now starch indicator, after adding starch indicator, what we find? It forms a blue complex of starch iodine complex. Okay, this starch iodine complex, which is blue in color. Now this is at the end point, almost very close to the end point. So this, if you add in the beginning, it gives a black brown color, and this starch being a carbohydrate, the iodine should form a complex with starch. It forms a complex, but if excess quantity, very large quantity of iodine is present, it will get trapped in the interstices, or the like lot of iodine is absorbed over the surface of starch. And what happens, this iodine, like when you get this color change from blue to colorless, उसके बाद ये जो एड्सॉर्ब्ड आयोडीन होता है, ये इंटरमिटेंटली बाहर, लाइक आपने शेक किया कोनिकल फ्लास्क, इट गॉट कलरलेस, ओके, ब्लू से कलरलेस हो गया. नाउ इफ यू हैड एडेड द स्टार्च इन द बिगिनिंग, दिस आयोडीन विल कीप रिलीजिंग. तो थोड़ी देर के बाद फिर से शेक करके देखेंगे तो थोड़ा सा blue tinge it starts appearing. That is, after some time the iodine adsorbed on the starch surface is released, and that iodine now imparts blue color to the solution. So then you think, अरे ये this is not the end point. तो फिर से एक और 1 or 2 drops you have to add, and then you check, yeah, now it is colorless. So such kind of interference due to addition of starch in the beginning occurs, and that is why starch is added to the solution only when iodine content of the conical flask is reduced and there is very little of iodine in that pale yellow color solution. होने के बाद starch is added. Okay, so what is the final reaction? Starch iodine blue complex with sodium thiosulfate, it gives NaI plus Na2S4O6 sodium tetrathionate, and starch which is set free is colorless. Okay, so these are the reactions taking place during determination of free chlorine. Things are clear. From here it is iodometry, same as that we have written the second part of in the DO determination also. From this, in DO determination, this iodine release is from, do you remember what is releasing iodine? Okay, let us go here. Now for this also you have to go for standardization. So potassium dichromate, you can see here a yellow colored solution, 10 ml is pipetted out. To this you add first a half test tube of dilute sulfuric acid. Here it is given a test tube with a marking here. So you have to transfer that much dilute sulfuric acid. Then you are supposed to add the 1/4 test tube of Ki solution to it and a pinch of sodium bicarbonate and keep it in dark. So when you keep it in dark for 5 minutes, then you take it out, you will find that the solution appears such dark brown color. Okay, so such dark brown colored solution, you have to first titrate with hypo solution till you get a pale colored solution. देखिए ये dark brown से ये pale हुआ. Now when this became pale, you are then adding starch to this. So when you add starch, it becomes blue like this, and then you titrate with it. You get formation of light blue colored solution. Are you getting what I am saying?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, so the first part of the experiment is standardization of sodium thiosulfate solution, for which you use 10 ml of 0.025 normal potassium dichromate solution, which was pipetted out and transferred to a conical flask. 1/4 test tube of 10% Ki solution and a pinch of sodium bicarbonate was added to the conical flask. Just a minute, I'll talk to you within 2 minutes. Huh?
मॅम तुमच्या व्हॉट्सअपवर
Okay, so we were at the standardization of hypo solution. 10 ml of N by 40 potassium dichromate was pipetted out and transferred to a conical flask. 1/4 test tube of Ki solution and a pinch of sodium bicarbonate was added to the conical flask. Shaken well, and then 1/4 test tube dilute H2SO4 was added, and then allowed to stand for 3 minutes in dark for completion of the reaction and to avoid photochemical oxidation of iodide to iodine. So we find that Cr2O72 minus plus 6I minus plus 14H plus gives formation of twice chromium 3 plus. Now because of this chromium, when you get a colorless solution, no, it is not exactly colorless. It has a light sky blue color to the solution, but definitely it changes from a dark blue intense blue color to a clear blue with a sky blue tinge to it. Okay, so this is the chromium form, and definitely the thrice I2 formed and 7H2. So this I2 is determined using thiosulfate, and towards the end point, when you are very close to the end point, you add starch to it, you get a blue color. This blue color will disappear when you titrate further with sodium thiosulfate to give a colorless solution where starch is set free and all of the iodine is consumed in NaI formation. Okay, so this is regarding the standardization. Okay, so titrate the liberated iodine with hypo solution from the burette. Towards the end point, that is when most of the iodine has reacted, the solution appears greenish yellow in color. At this stage, 1 ml of starch is added as an indicator. Which turns the solution blue due to formation of starch iodine blue complex. Titration is continued further till intense dark blue color changes to light green. Light green or light sky blue color generally is obtained due to presence of chromium 3 plus. Okay, Cr3 plus. So note, if Ki is not iodine free, a blank experiment has to be performed and the corresponding correction should be made to the titer value. Okay, so observation table, prepare an observation table for the standardization part. Volume of potassium dichromate, pipette volume was 10 ml, and then volume of sodium thiosulfate, that is burette reading. Like the first was 10.1, 10.3, 9.8, 9.8, and you will write the constant burette reading, which is the constant one getting, that is 9.8 m. So we will use this value to calculate the normality of thiosulfate solution. Nmol of dichromate is equal to N2V2 of thiosulfate. So that is how you will get N by 40 into 10 ml divided by 9.8 ml. So if you write it this way, then you get here 0.025 into 10 divided by 9.8. So this will be the thing. Clear? So that will be the normality of hypo. Okay. Okay. So this I think I have shown you. Yeah. Here you can nicely see that the color here is not colorless. This sky blue tinge, can you see a sky blue tinge to this part? And this
Yes,
is
ma'am.
due to chromium. And this sky blue tinge will be obtained only when you are titrating dichromate solution. If you are taking the water sample, chlorine water sample, it will be completely colorless. Okay, so when you are using this dichromate, this is the dichromate, then after iodine release is this, then you make it pale yellow. After pale yellow, starch is iodate, so this is with starch. And at the end point, when you add a thiosulfate solution, this dark blue color turns to this colorless solution, but it has a sky blue tinge to it due to chromium. Okay, so this is regarding standardization. Now coming to the second part, determination of free chlorine in the water sample. Now determination of free chlorine in the water sample comprises of, here we take 20 ml. So you are supposed to transfer 20 ml of water sample to a conical flask, and to this add a 1/4 test tube of 10% Ki solution. Now this is sufficient for 20 ml also, because this is 10%. 10% means quite concentrated. So that is sufficient 1/4 test tube. And titrate with, so when you add this, shake it nicely, you will find that yellow colored solution has formed. And titrate with hypo solution which is taken in the burette until the solution is pale yellow. Okay. Then add 1 ml of starch and continue the titration until the solution becomes just colorless at the end point. Note the confirmed titer value. What is the confirmed titer value? You have the volume of water. The observation table is pipette volume or the water sample taken is 20 ml every time. So first you took 20 ml and then you added these things and titrated with hypo. You added starch and the final reading was 5.5 ml. Then after the second reading gave reading 5.2. The third reading was 5.3. The fourth time you performed the titration, it is 5.3 ml. So you are writing the constant burette reading 5.3 ml in the CBR column. Now this reading we will use to calculate the free chlorine. So the calculation for free chlorine is free chlorine present in water sample is equal to normality of hypo into the volume of hypo upon the volume of water sample taken into 35.5 grams. So normality of hypo is something that you determined here. So this value will come here now. This N2 and volume of hypo is 5.3 ml divided by 20 ml into 35.5. So that will give you grams per liter or grams present in liter. So you multiply it by 1000 to get milligrams per liter. Things are clear?
Yes, ma'am.
Okay.
Yes, ma'am.
So the result is free chlorine present in given water sample was found to be dash milligrams per liter. So the calculation part is very simple. Then precaution. Pinch of sodium bicarbonate may be added during the titration of hypo solution. Then starch should be added when like this pinch of bicarbonate when you are titrating with potassium dichromate and a hypo solution. That time it is necessary because dichromate is there, then your H2SO4 is there and Ki is there. So due to light and all, and it may happen that the bicarbonate part is to make an atmosphere of carbon dioxide and excluding or it will keep away the oxygen in the conical flask. So that is the purpose of adding this. So the oxygen, atmospheric oxygen should not interfere or take part in any sort of oxidation reaction. That is the main purpose. Then starch should be added when color is pale yellow during standardization of sodium thiosulfate solution. You see, when you are doing standardization of sodium thiosulfate, you are using 10 ml of potassium dichromate. So you get a very intense, very large liberation of iodine, and there the color is dark brown. So in that case, it is very necessary that you first titrate with hypo solution and reduce the iodine content and then add the starch towards the end point. But in case of your chlorine determination, as such, the chlorine is very less. So you don't get a very large liberation of iodine. Okay, and that is why even if by mistake you add it initially, it will work. But with dichromate, you should not do such kind of thing. It is always mentioned that starch should be added towards the end point. Okay, so with this, the experiment completes. These are the photographs of this. Uh, this is actually your lab. You would have worked here in this lab, but then, okay. Things are
Ma'am, I'm
clear.
unable to get precaution one.
First precaution is pinch of sodium bicarbonate may be added during titration of hypo solution. Maybe is added during the titration of hypo with hypo. This is sodium thiosulfate. Now this sodium thiosulfate solution is not a primary standard. So you standardize it using another oxidizing agent. So the oxidizing agent that is used here is potassium dichromate. So this potassium dichromate will oxidize iodide to iodine. So this is not a direct oxidation of this. Okay. So what you are doing is potassium dichromate, which is an oxidizing agent, oxidizing iodide to iodine, and the hypo solution or sodium thiosulfate is capable of reacting with iodine to form NaI. Okay, so sodium thiosulfate can form or can react with the liberated iodine or use this liberated iodine. So how much sodium thiosulfate will be required, as much of iodine is released. Now you want to know how much iodine is released, as much of dichromate is present, right? Now apart from dichromate, if any other reaction is leading to formation of iodine, you want to nullify those reactions, right? So when you want to nullify those reactions, first precaution is you are adding sodium bicarbonate. So by adding a pinch of sodium bicarbonate, what happens? When this sodium bicarbonate is added, Ki is there, and you add dilute sulfuric acid. Now this is much more stronger and larger in quantity than the pinch of sodium bicarbonate, and you are sure that carbon dioxide will form. So the conical flask, inside the conical flask, effervescence will be seen, and the conical flask will have carbon dioxide atmosphere and the oxygen nitrogen present in air that will be moving out of the conical flask automatically, right? So that maintains the carbon dioxide atmosphere inside the conical flask. Okay, then another thing is why do you keep it in dark? Again, when acid is there, iodide is there, it may lead to by itself. Why this statement is written? Somewhere I saw this, no one asked me. This, if Ki is not iodine free, a blank experiment has to be performed and the corresponding correction should be made to the titer value. Now, इसका मतलब क्या है? When you prepare a Ki solution, generally we add like some quantity of H2SO4, and if you keep it for say 8 days or 15 days, बॉटल में बंद करके रखा है, फिर भी वो धीरे धीरे पेल येलो होते जाता है. It starts becoming yellow. That is all because of photochemical reaction leading to formation of iodine. Small quantity of iodine is formed. So Ki is never stored in a colorless glass bottle. Ki solution is always stored in amber colored bottle so that light will not interfere and photochemical oxidation of iodide to iodine should not take place. लेकिन उसके बाद भी अगर 15 दिन के बाद आप बॉटल खोलोगे तो it will be having a slight yellow tinge. 6 महीने के बाद खोलोगे तो येलो दिखेगा. Okay, so that is the reason. So these are some of the reactions that will add to iodine from other routes. One is photochemical oxidation, another one is the atmospheric oxygen present inside the conical flask may also interfere, and that is why this is done. Dark room, dark cupboards are there. Okay, small cupboard, like just below the platform you had photograph. Okay, you can see this is the like this is one table. This is first table. This is second table. So below that table in the lab will be cupboards. So you have to keep your conical flask in those cupboards, small cupboards which are there below the table. Okay, so that is the dark place. Things are clear? Any doubt? So readings are given or not? Readings are simple and the calculation is also very simple. So there is no issue in these things. So everyone got the thing?
Yes, ma'am.
Yes, ma'am.
Any
Yes, ma'am.
doubt? Okay.
Yes, ma'am.
So just a revision. Now all the 4 experiments have been explained to X1, X2 and X3 batch. You will be having this one also within no time now. So all 4 experiments from my side I have explained to you. If you have any doubt, you can ask me anytime. Tomorrow morning we have a theory class. Whenever there is any doubt, doubt will arise only when you will read for exam. Okay, so please read for exam, and you have to submit the PDF single PDF of 4 experiments on 17th, and you will appear for 4 batches will be appearing, Z1, X1, X2 and X3. I told you evening 6 to 6:20, time will be less because there are only 15 MCQs and every question will be given hardly 1 or 1 and 1.5 minute. Okay, so you should read because 4 experiments only are there. What questions I should set? So you say
Ma'am, so questions will be theoretical or solving?
MCQ question. Calculation also can be there. No, you have calculators with you. You can use
Is
scientific calculators.
JEE level or what?
Hmm?
Like what do we used to solve in JEE or what?
You used to solve in JEE. Yeah, something
No,
like
ma'am,
that,
that
but
kind
not
of questions.
that type of like you will have A, B, C, D option, and you have to select one of them. MCQ means multiple
Ma'am, so
choice.
information or numericals?
Yeah, calculation may be there, no? So you can use rough paper and calculator. एक्सपेरिमेंट में क्या क्वेश्चन पूछेंगे आप बताइए आप क्या क्वेश्चन पूछेंगे if I am student you are student what question you will ask me yeah that will be there clear yeah I am not getting any response from any student okay
I have doubt about uploading.
अपलोडिंग ना आपको बैच वाइज असाइनमेंट दिया जाएगा दैट टाइम यू हैव टू अपलोड ओके
Okay, ma'am. Okay.
बैच वाइज रूम बनाया है अपना अपना बैच में आपको अपलोड करना है आई गिव यू दैट आल्सो लिंक आल्सो
Okay, ma'am.
okay so by 17th you are submitting your file and 18th you are writing your test MCQ test 15 marks and 19th now I intend to complete the portion for your sessional so I may not engage the practicals because 26th is holiday so 19th को आपका viva होगा in on this 4 experiments okay so for viva 1-to-1 viva will be there like one student one teacher and during this 2 hours time whatever is given to you 1-to-1 or 2 students will be admitted at a time and the viva will be there 3 students at the max so viva will be for 15 marks so I'll be having like evaluation done for 30 by 21st x1 का 19th को होगा x2 का 20th को regular time 2 to 4 and for x3 21st को 2 to 4 okay so that will be your viva time any question from any other batch student are they really present
Ma'am, this Viva will be based on only experiments.
only on these experiments the calculation part the theory part like all those things totally based on experiment
Okay, ma'am.
okay then your theory MCQ test is also waiting first let me complete this by 21st then you will start reading for your theory exam so a 10 mark MCQ test I will give you there also so that may come up by 28th or so that will be announced because in theory we have covered water treatment and we will tomorrow and after tomorrow we will complete water treatment numerical part is almost done so only refractories adsorption and kinetics is remaining so two theory class I'll need for that that finishes the syllabus for sessional so by 28th if the portion is covered I can have an MCQ test on 10 marks MCQ test for the complete X section okay because at least out of 40 at least 10 marks should be done before sessional things are clear that is why I am hurrying up regarding your practical evaluation so that we can and you also can focus on your theory subjects. Clear? So should we close the meeting?
Okay, ma'am.
Ma'am, can you share this PDF now?
Now I'll try if it's possible. Yeah.
Okay, ma'am.
Okay.